Welcome, everybody. We're here for another presentation of the Living Earth Collaborative series of talks and the Department of Biology, the Group of Ecology and Evolution, Population Biology. Today, we have with us my graduate student, Vincent Fasanello. The title of his talk is going to be Microbes to Mountains, Why Dispersal Barriers Are Higher in the Tropics. And I'm sure that we're in for a really fascinating talk. Vince is a fantastic student that has been with me for 5 years already. Who would have thought that, Vince? It's a long time. Probably yeah, it's a really long time. And Vince has always been one of those fantastic people to have around in the lab. Intellectual contributions have been amazing. He's always been a really engaged person inside the department and among the group of graduate students. He came to us from Bucknell University, where he was a very active undergraduate researcher with Mark Hausman. And he did a lot of different things before coming to WashU. And I have to admit that when he first came to WashU, I don't think you were interested in the Botero lab necessarily. But through one of those rotations, it really became clear that both Vince's interest and his personality fit really well within the group that I was building. And I'm one of those really lucky people that can count Vince as part of the team members. I really feel very, very lucky to have him with us. So without further ado, I hope you guys stay with us for this fantastic presentation. And Vince, please take over.
All right, thank you, Carlos. Today, I'm going to be telling everybody a little bit about my PhD research in the Botero lab. The title of the talk is Microbes to Mountains, Why Dispersal Barriers Are Higher in the Tropics. And the work I'm going to talk about is inspired by a 1967 paper by Dan Jansen that some of you might be familiar with. It has thousands of citations at this point. In this 1967 paper, Jansen presents really a wealth of observations about global variation in dispersal barriers. But the main focus of the paper is really on thermal barriers to dispersal. And Jansen argues that mountains or other topographical features present really a physiological barrier to the movement of organisms rather than a physical barrier. And just to note, when I say and when Jansen says more or higher, when the barriers are higher in the tropics, he really means that they're more effective at limiting dispersal. So I'm going to walk through the basics of this hypothesis in one kind of animated slide here, and I'm going to use this as a roadmap for the rest of the talk. So we start with three really basic assumptions about the world. The first two we're going to go through are things that all of us have probably experienced in our lives. The first is just that temperature decreases with altitude. So this is something that you can even see in a single landscape. You can really easily see that lower elevation sites tend to be warmer than higher elevation sites. So here we see this low elevation valley relative to these really snowy peaks. If you ever lived anywhere mountainous, this is really easy for you to believe, probably. Temperature decreases with altitude. And yes, like I said, most of you probably experienced this in your lifetime. Maybe if you go on a skiing vacation or anywhere where it's warm and you can still see snow on the mountains in the distance. So our second assumption is, again, a really simple one. It's just that temperature seasonality increases from the tropics towards the poles. Now, if you imagine a tropical summer, you could picture a nice sunny beach. And in the winter, you'd picture that maybe it looks pretty similar. It's also nice and tropical and sunny. The temperate summer looks pretty similar to this sometimes, and in some places it's warm and the deciduous plants have leaves and it's generally pleasant outside. And then you also have the winter. So the contrast here is that there's really noticeable seasonality where you see this same landscape look very different and experience very different temperatures from month to month across a single year. So likely that you've noticed this as well, that there's temperature seasonality places where you've lived. And you probably also noticed that if you move from north to south in this country, or you go to a more tropical location, you could really notice that there's actually global variation in how much seasonality there is. And if you move a relatively short distance, you can experience this. Now, the third assumption here is one that some viewers might be less familiar with, but would be something that most evolutionary biologists and ecologists that are probably watching this are very familiar with. And this is just that populations adapt to the environments they experience. Now, here's just a fun example of a fennec fox. This is an organism that's adapted to a very hot environment. It has just a couple of examples, really large ears to help it dissipate heat. And you can't see them in this photo, but unlike most other animals in this group, it actually has fur on the toe pads of its feet, which helps it avoid burns from the sand. So this is an example of adaptation to the thermal environment that this organism lives in. And you can also really look at this topic with microorganisms. And here's just a picture of some yeast growing on a petri dish, but there are lots of different microorganisms you could study this in. Okay, so these are our assumptions, and we're going to use these now to build a set of predictions about global variation in dispersal barriers and kind of the consequences of that variation in dispersal barriers. This hypothesis goes beyond there to talk about some downstream consequences as well. So we're going to break this up into a tropical half and a temperate half, but let's focus on the tropics first. So here, and I'm just going to highlight on the side the assumptions that are relevant to what I'm talking about as I talk about it. That's what you'll see flashing by over here. But here, we can just see that for any given site, there's not very much variation in temperature from month to month across the year. And that's what's being depicted in this little plot here. And this is in the tropics. Now, we can couple this with our assumption that there's a relationship between temperature and altitude such that temperature decreases with altitude. And here, we might predict that temperature will be higher for a low altitude site, which is this site here near the top of our plot where the temperature values are higher. And it might be colder for a higher altitude or a higher elevation site like a mountain peak. And so this is the lower line on the plot right now. Consequently, there shouldn't really be very much overlap in climate between these sites at different altitudes. So if we take this kind of together with our knowledge about organisms and their evolutionary histories in the athermally seasonal tropics, we should assume that those species would have relatively narrow thermal tolerances. They would exist in maybe just one of these bands, either that high altitude site or that low altitude site that's being shown. And it's unlikely that they'd be adapted to thermal conditions that were outside of this range, just because they simply haven't experienced them. So the lack of climate similarity in space, coupled with the narrow thermal tolerances of organisms that live in the tropics, should work together to both result in increased dispersal costs when moving across a climate gradient or moving between suitable or similar sites in the landscape. In turn, this can result in reduced gene flow and ultimately high allopatric speciation in the tropics. Now, this explains all of the tropics. We're going to compare this and contrast it with the temperate zone. We're going to look at some predictions here, and it's going to follow exactly the same scheme. So in the temperate zone, there's obvious temperature seasonality throughout the year for any given site. So this is, again, I think this is our low altitude site. And we see that every month of the year, the temperatures are really different. There's a winter and a summer time, and the temperature band is much wider than you saw at the top. But temperature still varies with altitude. So again, the lower altitude sites tend to be warmer, and the higher altitude sites tend to be colder at any point in the year if you were to look at the same month. So consequently, there's notable overlap in the thermal regimes across the year for sites at different elevations. And you can see this with the little brackets. And from this, we predict that organisms have broad thermal tolerances simply because if they lived in either of these sites, they would need to adapt to survive in these conditions. And that would either be through actually enduring the conditions or some kind of avoidance behavior, which would be hibernation or migration or something similar to that. But we're going to consider mostly species here that would endure these conditions. That's where the hypothesis is most applicable. So our climate similarity in space, coupled with these wide thermal tolerances of temperate species, should result in decreased dispersal costs when moving between suitable or similar sites in the temperate zone. And this should really facilitate gene flow and allow populations to stay connected, and therefore will result in lower allopatric speciation rates. So there are lots of consequences of this kind of broad scheme that I'm talking about here, but one of them is the latitudinal biodiversity gradient, something that many of you might be familiar with. This is like a subsequent exploration of Jansen's hypothesis as like an exploration for this gradient. And this is just the idea that there are more species in the tropics. And you can see how this follows naturally from the scheme that we just built. If there's lower gene flow and higher allopatric speciation in the tropics, we'd expect there to be more species there than in the temperate zone, where on average you would expect higher gene flow and therefore more connectivity in your populations and less opportunity for allopatric speciation. So now that we have kind of the roadmap for the whole hypothesis and one reason why it might be an interesting hypothesis to think about, I'm going to tell you a little bit about two projects that touch on kind of different aspects of the hypothesis. At the end of the talk, I'm going to tell you a little bit about an ongoing biogeography project that I'm working on that looks at the variation in actual dispersal costs in the temperate and tropical zones. And in this project, I'm really exploring whether or not global variation in the magnitude of dispersal barriers follows these predictions that the barriers should be more effective at limiting dispersal in the tropics. But first, I'm going to describe an experimental evolution project. So this very bottom left-hand corner of the figure here, in which I explore the assumption that populations adapt to the environments they experience. And this is an incredibly broad topic, but I will specifically focus on the consequences of evolution in environments defined by constant versus variable stress exposure. So just as a little bit of an aside, first, just what is experimental evolution? Well, it's pretty much what it sounds like. It consists of just setting up a system in which evolutionary processes can be evaluated rapidly and under really controlled conditions. And so some examples that you might be familiar with would be work that's done in fruit flies. So that's the bottom left figure here. There's algae that people study this in, it's the top left, E. coli in the top right, and the organism I work with, which is yeast, in the bottom right. Although there are many other organisms people use for this, and even some larger organisms and organisms with longer generation times are used for experimental evolution, but these four are the main ones. Okay, so in a typical evolution experiment, you would evolve organisms in a range of experimental conditions, and you would subsequently quantify their performance in either those same conditions or in alternate contexts. And how you would do this would be with a fitness assay. And I put at the bottom of the screen just what fitness means here. It can really be measured in either of these contexts. But in essence, this assay is just to either directly compete lineages that you've evolved in different conditions to see how they fare against one another. Or if you have many different lines that you want to know how well they're doing relative to one another, sometimes it could be more efficient to actually assay them against some common reference line. And that way you know how well each one performs against a common benchmark. Now, I use yeast for my experimental evolution research. And this is just the same yeast that you would use for winemaking, baking, or brewing. There's really not anything different about the yeast I use. The only difference is that I've inserted genetic barcodes into the genomes of the lineages that we use in our projects. And these barcodes, which I'm just kind of showing in the right here, are just short genetic elements that are inserted into a neutral location of the yeast genome where they don't have an effect on fitness and they're not targets of selection. But what they essentially do is they serve as name tags, and they allow you to use genetic sequencing to rapidly census the relative proportions of lineages in mixed populations. So here I replaced the name tags with actual yeast-related names. And essentially, you would just be censusing a population to see how many Francine vitis are in it, or how many Pamela hops are present, and what their relative proportions are. You can do this really rapidly through genetic sequencing when all your lineages are marked with a very noticeable and easy to recover genetic tag. So this is kind of familiar to meta-barcoding for ecological samples. If that's something that you're familiar with, it's a tangentially related technique, and it has kind of a similar goal. It allows you to do a community census in an efficient and rapid way. And I'm not going to get deep into the details of our particular barcoding system here, but we have spent a lot of time working on one and developing one that's perfect for what we're trying to study. And you can read all about it if you want to in this recent publication, where we describe the system and we highlight its advantages for studies just like the one that I'm about to describe for you here. And if you see me like flip back and forth quickly between slides, it's just because sometimes my notes don't show up right away. Okay, so first I'll go through the design for this project now that we have all the background out of the way. And this project does follow the typical kind of experimental evolution project design. I'm gonna walk you through that with a little cartoon. So what we do is we start with 200 barcoded yeast strains. These yeast strains are identical except for these genetic barcodes. And they all come out of a little freezer box, which is this little gray square. Now we array these yeast lines, these 200 lineages, across 7 different treatments that are variable for chemical stress concentration and for the dynamics of that chemical stress. And I'm just going to quickly walk you through examples of those treatments. I'm not going to spend a lot of time dwelling on them here. We have 3 treatments that have constant stress exposure, and this is chemical stress that they're exposed to, and they're exposed to either 0% of the lethal limit, so this is their complete media environment. It's just the nice, pleasant environment for yeast to grow in without added stress. There's a treatment that's in that environment plus 40% of the ancestral lethal limit of some chemical, and then again plus 80%. So increasing constant amounts of stress as you go from top to bottom there. And then we also have 4 treatments that are in different regimes with daily alternating chemical stress exposure. So just as an example, the very top one in this box, this 0,40% ancestral lethal limit, this is the evolutionary history that just has every day has either alternated between the CM environment, so the no added stress, and then an environment with stress added at 40%, that ancestral lethal limit. And that's how you read all of these. The name of the treatment's a little code for what it is. So like EH4080 experienced the 40% and the 80% alternating. These names will come back up, but I'll introduce them again. So, and again, I said this was in chemical stress. This whole design was conducted in sodium chloride as a chemical stressor. I'm probably going to refer to this as salt as I go through here if I slip up. And also separately in copper sulfate, and I'll probably just call this copper because it's a mouthful to say copper sulfate. Okay, so once the lines were assigned to their treatments, yeast were evolved for 50 days, which is approximately 500 generations in this particular design. And despite this kind of cartoonish oversimplification, each one of these treatments actually consisted of 32 barcoded lineages evolving in parallel in the same environmental conditions. So that we had many, many replicates of essentially what's going on when you evolve in those conditions. Now, at this point, I've mentioned evolution several times in the context of experimental evolution, and it might seem a little bit ambiguous. It's actually really simple. Evolution here is just culture with serial dilution. And here's a less cartoony diagram of what that is. If you were to look at kind of that top left portion, that would represent a saturated yeast culture. This is yeast that's grown in an environment until it's already used all of the sugar that's there for it to reproduce. And it goes into a stationary phase where it essentially sits and waits to be introduced to an environment where there's, again, media for it to consume and to reproduce with. So every day you dilute this yeast that's grown overnight for 24 hours. You dilute it 1 to 1,000 or through some other relatively extreme bottleneck into fresh media, and you let it grow again. And you just repeat this over and over and over. And in this system, each one of these transfers is 10 generations. So here, 50 transfers is 50 days is 500 generations. So at the end of the 50 days of evolution, where all of our lineages have been evolving in their different treatments, we then actually pool all of the lines from all 7 treatments from a single chemical, so either the salt treatments or the copper treatments, and we pool them all into a single population, which we're going to then kind of simultaneously measure fitness in relative to a reference. The pool is spiked with the reference strain, which was represented by this little gray circle here. And you put in this reference strain, which is just, again, like I said earlier, kind of a benchmark to measure fitness against. This is added into your population, and then you allow it to grow for 2 transfers, so 20 generations. And with that knowledge and a little bit of math that I'm not going into here in detail, you're able to calculate the fitness of each strain based on how it performs over 20 generations relative to that reference that you've spiked it with. Now, we repeat this exact process with the freezer stocks from before the experiment began in order to measure the initial fitness of the lineages before they were exposed to any of the different treatments. And here we expect that there's not going to be very much difference in fitness between the different lineages. Anything you would measure is essentially noise. And this allows us to kind of measure change in fitness over the 2 time periods. And fitness is context specific. So we measured fitness at day 0, so this very first column here, and then at day 50 in 2 different environments for each yeast line. We measured them in a no stress added environment, which was the complete media, and in an environment with chemical stress added. And so that's complete media plus either salt or copper. The lines were all assayed in the type of chemical that their treatment block evolved in. None of them were crossed between chemicals. So like I said, from these 2 different fitness values, the one at the beginning of the experiment and the one at the very end of the experiment, we're able to measure the change in fitness over 50 generations for each lineage in each environment. And this is the response variable that you're about to see in every single one of these plots that comes up for the results for this section. So now that we understand a little bit about experimental evolution and the design of this particular project, I'm going to jump into answering some sub-questions within our broader topic of the consequences of evolution in variable versus constant environments. The first question is a really simple one. It's just asking, are there trade-offs between fitness in the stress-added environment and in the no-stress environment? And I'm going to walk through what this means. So we're going to see some plots that look like this. On the x-axis of these plots, you see fitness change in some environment. And on the y-axis, you're going to see the different treatments. And so here on the bottom, we have this EH0. This is our treatment that evolved in complete media, so the no added stress. And at the top is the EH80. You can think of this as the line that evolved in the highest constant amount of stress. So we're interested in looking at trade-offs. We want to assay them in these 2 environments that they evolved in. So at this top panel that I'm showing you right now, we're plotting fitness in complete media. This is the no stress added environment. And this is for the treatment involved in that environment, EH0, and the one all the way at the other end of the spectrum, like I just said, the EH80 treatment. Now, the second panel, it has exactly the same layout as the first, but here we're looking at fitness in the environment of the EH80 treatment, so the one with the brown line pointing to it. So I'm going to walk through what we might predict to see here, depending on the different evolutionary outcomes that could occur. First, if there's no adaptation in our 500 generation experiment to either of these environments where these yeast lines evolved, we'd expect to see no fitness change. So no deviation from being right centered on this 0 line for either treatment in either of the conditions. If the treatments adapted to the environments they experienced and this didn't have a trade-off in the other environment, so if it didn't trade off with performance in the alternate environment, we'd expect to see a situation like this one, where our EH0 treatment increases significantly in fitness in the environment it was evolved in and shows no change in fitness in the alternate environment. And then the alternate is true for this EH80 treatment. It significantly increases in its home environment, but again, doesn't change in fitness in the alternate environment. Now you could imagine a situation where there are trade-offs, and this is just exactly the same as this last slide, except now when you're assayed in the alternate environment, so the EH0 treatment, which evolved in complete media, being assayed in complete media with added stress, you would expect it to decrease in fitness there. And the same for the evolutionary history that evolved in the high stress, you would expect it to increase in its home environment and significantly decrease, so be significantly less than zero in the alternate, the complete media environment. There's a couple other options that we could see here. We could see that there could be mutations that have positive pleiotropic effects, and this would just mean that adaptation to one environment also has benefits in the alternate condition. So here, adapting to complete media gives you a benefit in complete media with added salt, and adapting to complete media with added salt also has translating benefits to your fitness in the environment without that salt added. And then finally, there's a whole litany of different kind of more complex cases. This is just a simple one that I want to lay out because we are actually going to see it in a second. This is a case of asymmetrically positive pleiotropy. And it sounds like a complicated thing, but basically what we're seeing here is that adaptation to the complete media environment, so the greener box here, without that added stress, provides a benefit in the presence of stress. But evolving in the presence of stress does not provide a benefit when you're assayed in the absence of stress. And I know that's kind of a mouthful, but you can see it up here, in that this one in its home environment increases in fitness, and it increases in fitness in the alternate environment, and that the same is not true for the evolutionary history 80 treatment. Now I'm going to make this a little more concrete by showing you some actual results. Here we're looking at the SALT data set, and we see that there are trade-offs evident. So I'm just going to flash this cartoon for the trade-offs back up. This is what trade-offs we expected trade-offs to look like, and this is what we observe. So here is our treatment that evolved in the CM environment, decreases in fitness in the environment with added stress, so not the environment it evolved in, but increases in fitness in the environment that it did evolve in. And then the alternate is true here, where the lineages that evolved in the environment with added stress increase in fitness there, but decrease when that stress is not present. So like I said, we also did this in copper with copper stress, and the results for the copper sulfate data set are different from what we saw in salt. Here we see kind of that more complex asymmetrically positive pleiotropy case. So that's the one that looks like this. Where evolution in the no stress added environment results in elevated fitness in the presence of stress, but evolution in an environment with stress doesn't confer fitness benefits when that stressor is taken away. There's that cartoon again. So to answer our first question, the existence of trade-offs between environments on opposite ends of a stress gradient, so a place where that stress isn't present all the way up to a place where it's present at a high concentration, it's really context dependent. Sometimes we see trade-offs, sometimes we don't, and that depends on the stressor, the type of stressor, and the mechanism of adaptation. Something else we're interested in, as we mentioned, we had 7 different treatments here, but that we so far we've really only looked at the 2 most extreme ones. It's possible that there might be some relationships in the center that look a little bit different. Maybe we don't see this trade-off there. So looking at treatments that had either fluctuating evolutionary histories, so they were the daily alternating ones that alternated between 2 different contexts, or there was one that was an intermediate and constant value also. So this is the same data we were just looking at. It's exactly the same plot. I've just expanded the y-axis on both panels to make space for these other treatments that I'm going to plug in. The treatments, this is just like a little insight here on the right. The treatments are ordered by the amount of stress exposure in their evolutionary history. So the maximum and mean stress, which kind of holistically can be thought of as stress exposure, increases up the y-axis. And it's ordered this way just to aid in some interpretation in the following slides, because we see that many of our patterns really follow this ordering. Okay, so here's all the data. We have not just these 2 treatments, but data for our 5 other treatments as well. And this is for the NaCl data set. On the left, I'll show you the copper data in a minute. And as before, in the top panel is the fitness change in complete media environment, and the bottom panel is the fitness change in that same environment with added salt stress. Now, I'm just gonna box in the results from before. These are the trade-offs that we saw. And I want to show you that the same results extend to the more intermediate and fluctuating treatments. So anywhere where I've extended the box, those treatments have the same relationship to zero as that most extreme treatment that they're close to. So in other words, treatments with more stress exposure, those that are near the top of the y-axis, perform similarly to that EH80 treatment, so that the top stress treatment, the one with the highest constant stress. And treatments with less stress exposure, so those near the bottom of the y-axis, perform similarly to that bottom treatment, that EH0 treatment, which grew in the complete media without any stress during its evolutionary history. And like I said, we have the same data in copper. And remember, in copper, we saw that weird asymmetric positive pleiotropy. And here's that just highlighted for you again, just so you can see that those results are still here. And again, we'll extend out the results to show the neighboring treatments that share that same relationship. And just like in the left panels, we see that intermediate and fluctuating treatments, so all the ones between the two extremes, really perform similarly to the extreme treatments they're closest to on the y-axis. There's some indication that they perform closer to the higher stress side of the axis that they're close to. But there's really no evidence, if you look both at the copper, so the right-hand side, and the salt, the left-hand side, there's really no evidence that the intermediate or fluctuating treatments, so all the ones that aren't at the top of the bottom or the bottom of each box, there's no evidence that they're qualitatively different. It looks like they're mainly intermediate. So they're not mitigated. We're gonna say the relationship that we saw between the extreme treatments really isn't different if you look at evolution in fluctuating or intermediate conditions. Instead, it's different only in amount, not really in kind. So another thing we were interested in kind of beyond this, taking into account that we had these different relationships with stress and our two different chemicals, we wanted to know whether it was dynamics or exposure that shaped fitness in the CM environment or the environment with the stress added, kind of by directly comparing the treatments rather than comparing them against zero, since we noticed they had such different relationships to zero. We wanted to see if we could find maybe a common theme that tied together evolution in these constant and stressful environments versus fluctuating and stressful environments. So again, I'm going to build off of this same underlying plot. We're looking at these same 7 treatments, the same data for salt on the left here. But now I'm using stats to compare fitness between the groups, and that's the colored boxes here. Colored boxes indicate significant relationships, and the numbers in the boxes are the estimates for those relationships. I think it's a little bit hard to interpret these stats, and it's very hard to interpret them quickly. So I've translated them into rankings that I think will help discuss this. So here on the kind of the left-hand side, you see the rankings where, if you look at panel A, EH0 is rank 1. That indicates that it is the best performing or the highest fitness treatment in that environment. So in this NaCl data set, we see that the rank in this complete media environment, so that's our panel A, for all of our treatments is negatively associated with the amount of stress exposure in evolution. And we see that completely opposite to that, the rank of fitness values among these treatments in an environment that has chemical stress added is positively associated with that degree of chemical stress exposure in evolution. And again, there's not really a major difference between fluctuating and constant treatments. So none of those middle 5 treatments that are in the intermediate section look different from the extreme 2, other than just kind of being intermediate. Now we have, like I said before, these same results in copper. And again, you already saw all this data. But now here are the stats for comparing amongst the copper groups rather than relative to that dotted 0 line. And for ease of comparison, here's the rankings in the copper sulfate. And we can see that it's really similar to what we see in panels A and C. Rank in the CM environment, so the no stress added environment in the top right-hand panel, is negatively associated with the amount of stress exposure in evolution, which makes sense, and rank in the stress added environment, so the bottom right-hand panel, is positively associated with this degree of exposure. So if you remember, there were different dynamics relative to zero in these two treatments, where we saw trade-offs in salt and the asymmetrically positive pleiotropy in our copper data set. But we find that even though there are really different mechanisms of adaptation, we see that the rankings in the no stress added environment, if you compare between panels A and B, so compare the salt and the copper data, see that the rankings are very similar. In both cases, it's the treatments with less stress exposure in their history that perform better in this environment that has no added stress. And we see the same thing if you look at panels C and D, which are looking at fitness in this stress added environment, where rankings in this stress added environment are similar in the NaCl data set, so C, to the copper data set, so D. And here it looks like higher stress exposure outperforms the treatments with lower stress exposure in their evolutionary history, which again makes sense. So from this, we can conclude that it's really the degree of stress exposure in evolution that shapes fitness in the CM, so the no stress added environment, and in that environment with added chemical stress. It's not actually the dynamics of exposure that appear to be the most important factor. And we see that this is true regardless of whether or not there's underlying trade-offs in fitness between the environments. So that's kind of taking together A and B here. Now, finally, for this section of the project, we ask whether evolution and fluctuation itself is costly. You could imagine that it might be. Lineages evolving in daily alternating conditions would experience pressures to adapt potentially to two different environments that could have different selection pressures. There's also a chance that the switching itself, so nothing having to do with the particulars of the environments, but the actual switch, could also be a target of selection, further increasing the number of selective pressures on the kind of lineages selected for generalization, so the ones from fluctuating or intermediate histories, versus those that experience those extreme constants, so the no stress or the high stress added environment. So I'm again going to walk you through a setup for a different kind of plot here. This is also looking at fitness change, but now we have fitness change in the no stress added environment on the x-axis and fitness change in the environment with chemical stress on the y-axis. And we're going to see that the points for lineages and the points for the treatment means appear at the intersection of their fitness values in this xy coordinate system. So just to kind of walk you through a cartoon example, this is the fitness change we might see in a CM specialist. So a set of lineages that was actually evolved in this x-axis. You see they moved positively in fitness change in the x-axis dimension, but negatively in the y-axis dimension. And you can see for a specialist on the other side, so this would be our EH80 treatment that evolved in the complete media with the NaCl stress. You see that this treatment would be predicted to increase in fitness here, so on the y-axis, but decrease in fitness along the x-axis, so be lower than 0 on the x-axis. Now, we can connect these two treatments with a line that kind of represents our expectation if there was no cost to generalization. In other words, this line kind of represents the expectation if generalists, so lineages that were evolved in fluctuating or intermediate conditions, if they differ again only kind of in amount, but not in kind from the specialists. They're just adapting to a different amount of stress, but not doing it in any kind of different way or suffering from other different pressures. Now, like I said, lineages and treatments with kind of joint performance values that fall on this no cost line, they're intermediate between the specialists, and we're going to see that they don't trade their mean performance for breadth of adaptation. That's something I'm going to repeat a couple of times here. And so it's important to kind of get straight in this, the specialists are not increasing their breadth of adaptation. They're actually decreasing their breadth of adaptation as they evolve for a higher mean performance, which is elevated by getting a large fitness gain in one environment, where you would expect that the generalists might have to hedge their bets a little bit. So the treatments that are in the middle may need to deal with both these environments, and again with the switch. So they might pay a cost, and we'll see what that means in a second. So lineages and treatments that pay this cost would fall kind of below this line, and they would trade their mean fitness for breadth of adaptation, like I just said. In other words, they kind of attain a lower joint performance in order to balance their performance in the two environments, or to also deal with this switching stress that exists. And you could also imagine that maybe there are lineages or treatments that fall really above this no cost line, and that they escape this trade-off and somehow enjoy an elevated mean fitness and an elevated breadth of adaptation. And you could imagine that mutations that are beneficial in both of these environments are relatively rare compared to mutations that increase fitness in one environment, or that increase fitness in one environment at the expense of fitness in the other. Now, this is just following exactly from our cartoon, but here are actual data. This is for the NACL data set. We can see that in all cases, our intermediate and fluctuating treatments, so all of these other larger dots that don't fall on the line, and again, they're in exactly the same order they were in all the other plots, they actually obtain breadth of adaptation at a cost to mean performance if we're following this scheme. And the data are really qualitatively similar for the copper data set, so just to save some time, I didn't put that in here. So here, we're showing that there is a cost of evolution in intermediate and fluctuating conditions. So lineages from these treatments really underperform the expectation for a comparable specialist, which you would have expected to fall on that costless line. But it's also really nice to notice here that there's a lot of spread within each of these treatments. So if you look at kind of these ellipses, they show, I'm pretty sure these are 85 or 95% of the points will be inside of the ellipse. But there are even points outside of kind of this confidence band. And maybe there's more that we can gain by looking at these individual points and thinking a little bit about them rather than about the treatment means. And so that's what this next slide is going to do. There's a lot on this one. I'm going to walk through each of these panels, at least for a couple seconds, so we can spend some time just looking at them. But just to orient you to what we're seeing here, this is the same data as the last slide. Panel A is the last slide. Panels B through H are breakout panels for individual treatments. So it's the same data, but I'm just plotting one treatment on each of these panels. So you can really see the shape that each of these treatments take. And let's see, okay, what are some things we can get out of here? First of all, we notice that there's lineages in each of these fluctuating and intermediate treatments. So C, D, E, F, and G on this plot. And I'm going to walk through those. And they fall on this costless line or above them, despite the fact that we see these treatment means, if you can see my mouse, in A that fall below the line. Now, these are small, so I'm just going to walk through some of these panels now and show you some examples. This is our treatment that evolved. If you look at the bottom right, there's a little key that I'm going to walk through. This is our treatment that evolved in the fluctuating environment that fluctuated between no stress and 40% of the ancestral lethal limit. You see, for the most part, the points fall below the line or in a similar location to many of the points in panel B, which is that CM specialist. But like we were just talking about, there's a couple points that fall far above the line that escape the cost and somehow obtain both breadth and depth of adaptation. This is also present in our constant intermediate stress environment. In an intermediate fluctuating environment, this one goes between 20% and 60% of the ancestral lethal limit. Here you see there's a good number of points that fall above the line. Here's an environment that alternates between the most extremes. So every day you would alternate between the CM environment, so the environment that B evolved in, and the most high stress environment, so the environment that H evolved in. And here you can see that there are lots of lineages that fall on this no cost line that escape this trade-off. And there are even some that seem to be better. And again, you see this in the last treatment. So there tends to be a cost of generalization, but this isn't universally true. So to conclude this section, and this was the longer section, the next section is relatively short. We find that fitness is shaped by the amount of stress exposure in evolutionary history, not the dynamics of that exposure. And that this observation doesn't depend on whether or not there are underlying trade-offs in fitness between environments. We also find that generalists tend to trade mean performance for breadth of adaptation, but there are some lineages that escape this cost. Evolution might favor these lineages in mixed populations over longer timescales. And now that we kind of have a little bit of a better understanding of how organisms adapt to fluctuation and fluctuating and constant environments, I'm going to return kind of back to our broad scheme and then talk about the biogeography project quickly. So here we are back at the broad scheme. We were just in this bottom left-hand corner. Now we're going to move over to the predictions half of the slide for this very last 10 to 15 minutes. Specifically, we're going to explore variation in dispersal costs between suitable sites within the tropics and the temperate zone, just like we were talking about way back at the beginning of the talk. And this project uses comparative phylogenetic and biogeographical techniques to actually quantify variation in the magnitude of dispersal barriers throughout the world. So we're really measuring the dispersal barriers themselves. I'll walk quickly through a simplified scheme of this design as well. First, just some really quick definitions. Here, dispersal barriers, since it's been a little while since we introduced them, are just unsuitable habitat separating sites in an environment. And when we say that they're higher in the tropics, we're really asking whether or not physiological barriers of the same magnitude are more limiting to dispersal in the tropics than in the temperate zone. So our goal for the project, like I said, was to measure variation in dispersal costs between suitable or similar sites globally. And here we have just kind of a mock-up of the world with a temperate site. So this is our blue site on the left, and a tropical site. It's a red site on the right. These are just two example cartoon landscapes that have some topographic relief. I'm going to use them to explain this a little bit more completely. We can put yellow dots on here, which represent starting points or the location where an organism currently resides. And we can add relevant ending points for dispersals. And those would be in environments that are similar to the starting location. So here, it's a valley on the other side of the mountain range. In this case, the dispersal barrier is the high elevation mountain pass separating the two suitable valley sites. If you imagine this mountain extends out kind of in the z-axis here as well. But we could also imagine a case where the topographical barrier is for a mid-elevation species. Here, the species would have a target that's another mid-elevation site for its dispersal where conditions are similar. And this organism might disperse over the mountain peak or through that low elevation valley. In both cases, the mountain or the topography itself presents a barrier. Now, finally, we could also even imagine a case where valleys are barriers to mountaintop species. These species would have to disperse among mountain peaks that could be separated by valleys, and the valleys between them here would be the dispersal barriers for these organisms. So this all seems nice and simple, but choosing appropriate starting and ending points in a non-cartoon world is not trivial, and that's the main interesting point here that sets this apart kind of from previous work on this topic. So our solution was to quantify dispersal costs between ranges, between locations in the ranges of avian sister species pairs. So sister species pairs just represent known recent speciation events. So they're species that are each other's most close common relative. And this is just a coverage map of our data set that I'm going to show some very preliminary results from. It depicts the pair centroids for 650 sister species pairs, which is approximately 1,300 species. And just note that they're distributed across latitude and that we have data for multiple realms. Although this may not be the final data set, it might get pared down a little bit. So now we're going to focus on the between locations part of the statement and talk about really quickly what this means. If we have the ranges for a sister species pair with range A in green and range B in pink, and you can see their phylogenetic relationship in the bottom left, we would want to quantify the cost to disperse from a location in the range of species B to an appropriate location in the range of species A, because these species are each other's most common relatives. Or from a location in the range of species A to an appropriate location in the range of species B. But we don't want to simply quantify dispersal cost or barrier magnitude over a route as the crow flies. So we don't just want to draw an arrow like this cartoon has done, but rather we want to make a route that minimizes the physiological cost along that corridor. And that might look like a very snaky route. It may not be directly a straight line. And to do this, we use a least cost path analysis. And again, I'm not going to get deep into the details of this, but briefly what this does is it allows us to construct paths between all locations in the ranges of species A, so the green species, and species B, so the pink species, and then retain only the subset of paths that minimize the elevational or thermal dissimilarity along the dispersal route between those two polygons. So skipping over a lot of details with this that you might get to hear more about if you tune into my next talk, which will probably be my thesis defense. This ultimately allows us to report a single elevational dispersal cost and a single thermal dispersal cost for each species pair. So here the sister species pairs are our unit of analysis, and we have a single value for the elevational dissimilarity along the route between them and the thermal dissimilarity along the best route between them. And we use these to estimate the barrier sizes. So I'm just going to present two of our predictions and follow up on these with preliminary results. But like I said, this is ongoing, and this project will probably have more at the end. So this is a little bit of a big prediction here, but I'm going to try and talk through it quickly. We have the idea that sister species represent successful speciation events. We know that they are speciated, separate species that are each other's common relative. And in the thermally aseasonal tropics, we'd expect that small barriers would be sufficient to allow speciation by preventing gene flow, just like we talked about when we went through the cartoon at the beginning, talking about the hypothesis. But we would also expect that large barriers are present still, because you could imagine that maybe there's still really high mountains and there are organisms that live in the valley. So you would have high and low peaks separating species in the tropics. And you can see that in this kind of cartoon. And then we predicted the minimum barrier size sufficient to allow allopatric speciation to occur kind of through this reduction in gene flow mechanism. We think that those are going to increase towards the poles because species in these regions have broader physiological tolerances, and dispersal across the landscape is less hindered by thermal stratification with altitude because of seasonality. So you would need a bigger barrier, essentially, to keep species from being connected. And our second set of predictions just explores kind of the relative strength of elevational and thermal barriers. So we consider whether latitude better explains variation in thermal or elevational barriers separating sister species. And we'd expect a stronger relationship between latitude and thermal barriers if temperature really is the primary factor responsible for dispersal barrier effectiveness. But if instead there's a suite of factors that kind of vary with altitude or vary with elevation that might be important, maybe it's temperature, maybe it's irradiance in addition to temperature, maybe there are lots of different things that kind of combine together, we'd expect a stronger relationship between latitude and elevational barrier magnitude, because those elevational barriers kind of capture a suite of different potential climatic factors that temperature is just looking at temperature. So this project isn't finished, but I'm just going to quickly pop up some preliminary results here. First, we're going to focus on elevational barriers globally as the top left panel. So here, there's just model results on the left, and then a plot of the figure with the fitted model on the right, and that's going to be the same for every panel. Here we see that elevational barrier magnitude increases from the tropics to the poles, and we see that there is an indication that elevational barrier magnitude might also increase from just generally from south to north. So it's greater in the north than in the south. And this supports Jansen's hypothesis. Now we can look at the same thing, but looking at thermal barriers globally. And here we see a very similar relationship. We see that thermal barrier magnitude increases from the tropics to the poles among avian sister species pairs. And again, this supports the hypothesis. And you can see the little prediction cartoons in the corners of these, although they're probably pretty small for you. You see we kind of match those predictions, at least roughly. Now, the right-hand side, we're restricting analyses just to the northern hemisphere. This is a little bit more of a conservative estimate. And again, we see the same results for elevational barriers when we look only at this hemisphere. Barrier magnitude sufficient to allow speciation increases towards the poles. And again, when we look at thermal barriers just in the northern hemisphere, so in the bottom right, we also see that the barrier magnitude sufficient for allopatric speciation increases as you increase in latitude. And if you remember back to that second set of predictions that we had, we talked a little bit about the fit, and we can see that latitude better explains variation in thermal barriers than it does in elevational barriers globally. And when we restrict our analysis just to the northern hemisphere. And this is the same thing with just some little polygons drawn over to emphasize the shape of the data in each of these different plots. And I'm just going to walk through the conclusions and then we'll end from here. But what we see is that overall, the magnitude of elevational and thermal barriers sufficient for speciation increases from the tropics to the poles. This supports the hypothesis. And we see that the minimum barrier size sufficient for this speciation also increases from the equator to the poles. And that's if you're kind of looking at the shapes of the points here. And this is because big barriers exist everywhere. And finally, we see that there's a better fit for thermal barriers than elevational barriers when we look at the two and kind of try and determine which one is better explained by latitude. And this all provides really broad support for Jens's 1967 hypothesis about why dispersal barriers are higher or more effective in the tropics. And with that, I'm going to conclude by saying that, again, this work is ongoing, but please keep an eye out for more on the experimental evolution and the biogeography projects in the very, very near future.
Vince, thank you very much. I hate to put you on the spot, man, but this has been such a wonderful talk that I'm like even choking up saying, ending this up. This has been an amazingly clear presentation of your work. It shows your breadth and your capacity as a student, and I'm really very proud for all the stuff that you've shown. Like, it's really very incredible work.
That means a lot.
There are a few questions in the chat, and Jess, even though I said that maybe you could take just the whole thing, now I have the YouTube chat open so I can help out with one. And I will start you off, Vince, with the first one. It's from Michael Moore, who asked the following question: how much does the delay between environmental fluctuations matter? If fluctuations occurred within generations or between sequential generations, would there have been broader tolerance in the fluctuating treatments?
Yeah, so this is actually a topic of a lot of interest, and we do think that we might have seen different results if the patterning of the fluctuation kind of lined up with the generation time differently. So for anyone who's not super familiar with this topic, basically, if you have fluctuations that occur within a generation or occur relatively rapidly, you might expect that organisms would need to adapt to both of those conditions. But if you had maybe slower change, so maybe in our condition here where you had 10 or 15 or 100 generations in a condition before it changed, you might expect adaptive tracking, which is more like the population kind of just follows the environment as it moves rather than creating generalists or having different strategies to deal with both of those things. And I guess we have an interesting intermediate here because the yeast, while they do get 10 generations every transfer, it's an oversimplification to kind of view it as non-overlapping generations. The same yeast cell could be producing offspring in every single one of those and could potentially experience two different environments. So there's a long-winded answer to the question, but basically, yes, the patterning makes a big difference, and that's something that we're really interested in.
All right, I don't see Jess here, so I'm going to take the next question as well. This comes from Yusan Yang. Do you think that the results that fluctuation of stress is not as important is something that is unique to chemical stress or generalizable to other types of stress as well?
Yeah, I think that, like many things that you look at in experimental evolution, there's going to be some context specificity and then something that's a little bit more general. I think that a big difference, maybe if you were just think of an example between chemical stress and maybe a stressor like temperature, is that, I mean, the optimal amount of copper sulfate stress is probably none. And so it's kind of a one axis where you have a really good environment, the benign environment with none of that stressor. And then your changes are just having more and more of it, or experiencing that more stress and different dynamics. Where, as you could probably expect to see potentially different outcomes, where in an environment where the stressor like temperature has an optimum, and where if you move either to the left or to the right of that optimum, both of those directions are costly, but maybe not symmetrically so. So maybe existing in slightly warmer environments is less stressful than existing in slightly colder environments. So yeah, there's a lot of opportunity to see either the same or different things depending on kind of how you set things up. I think you would expect to see similar outcomes in like this one axis stress, where you have like a good condition and a quotes like bad or a different condition. And I think this is relatively similar to some of the things that you would see with macroorganisms. If you think about them on kind of an evolutionary timescale, you would picture that the environments that these organisms have an evolutionary history in, and that their whole group has had a history in, maybe those environments are the benign condition for them. And then any more stress or less stress from that would be the stressful condition. Although, yes, there could be differences depending on the chemicals or the type of stress.
All right, there's two more questions, one from James Stroud and the other one again from Yusan. I'll take the first one and then Jess could help us close up. James asks, well, first of all, he says, great talk, Vince, good job. I wholeheartedly agree with that. Do you think that the tropical versus temperate patterns you found extend to other taxa?
I do. I think they do. I think the reason that we really started with birds was, well, one, we have a lot of data for birds in our lab, but two, we really thought that if the hypothesis wasn't going to apply strongly to something, it was going to be birds, because they have one of the most modal of the the higher organismal taxonomy you might think of. You're comparing mammals or aves or amphibians or reptiles. So we would think that if any organism was able to kind of escape and avoid measurement of this hypothesis, it would be birds. So I guess if I had to guess, I'd say I'd expect a stronger relationship in less modal organisms.
All right, great. Thank you, Vince. And thanks, Carlos, for facilitating those questions. We're running a little bit short on time, so I'm just going to ask another one of Yusan's questions, which is kind of a follow-up to what you just answered.
Sure.
And then we might have to stop there. But Yusan asks, related to the previous question, she was also wondering about that, perhaps related to whether the taxa is ectotherm or an endotherm. And are there other important factors that you could think, or other factors that you think could be important?
Yes, there definitely are. So the kind of following off of the ectotherm, endotherm idea, well, as an ectotherm, you would definitely be exposed, kind of more exposed to the environment you're in. You would have to be either at that temperature or close to it if you had some kind of avoidance behavior where you could avoid that temperature a little bit by being in a burrow or something like that. You could imagine that endotherms, since they're able to manipulate their body temperature, might be able to avoid a little bit of the costs in the same way. Maybe they could move through environments that were more dissimilar. But I think another way that you really see organisms get around this, having to deal with a really wide range of conditions, like you see somewhere like St. Louis, right? Like it's sometimes close to 100 degrees in the summer and close to 0 degrees in the winter. That's a really broad range of conditions to endure. Lots of organisms will disperse seasonally, so they'll have seasonal migration to avoid those conditions to which they're not really optimally adapted. Or some mammals, like a good example is a bear, would go through hibernation to avoid those really harsher parts of the year where maybe it's not the temperature per se that's important to the bear itself or the organism hibernating, but it would be to its food sources or other things that are important in that environment.
Great, thanks. So we'll end the presentation there. There was another question, but we'll make sure you get in touch with Michael Moore to answer his separately. But thanks so much, Vince, for your talk. That was fascinating. And I just want to remind everybody, if you're part of the Living Earth Collaborative, we're having a Zoom reception starting in a few minutes if you receive that link via email. And yeah, and thank you again, Carlos, for hosting. And great job, everybody.